Hello, my name is Rachel, and I'm also a part of the Spider Lab at NGRREC. I was lucky enough to be a research mentee under Dr. Brett Seymour, alongside PI Tony Dell, and then also with Onicia, Kate, Katie, who just presented, Arjun, and Stella, who came, helped us out. And this past summer, we've been looking at how various factors, abiotic factors, can influence foraging behavior or like predator-prey interactions across different temporal niches. And I specifically am going to present today on metabolic rates of wolf spiders. Circadian rhythm and metabolism. So circadian rhythm is a significant biological process that occurs in any organism, and this in turn regulates our metabolism, which is another important biological process that occurs in all organisms. And it's an internal process that's endogenous or originating within the organism that is entrained by the environment, which you can see what entrainment means, which is the process by which extrinsic stimuli can change or reset the phase of a circadian clock. And if you can see those images below and see how continuous darkness can change that phase of the circadian rhythm, which we see on the right. So while circadian rhythms are endogenous, they also can be adjusted to the local environment via like external cues. And a lot of important research is currently being done on how different external environmental conditions can affect and influence our circadian rhythm. So, what does this have to do with spiders? So, like Lycosidae or wolf spiders, okay, let me back up. So light is an important driver of these specific biological processes, and arthropods are entrained by light and temperature conditions, which can change throughout our 24-hour light-dark cycle. Lycosidae have two distinct temporal niches, which are nocturnal or diurnal, but they also can be considered, let me scroll down for my notes. Nope. So they also can be considered active during dawn or dusk if it's a lot of heat. And evidence has been shown, like Katie mentioned, that spiders don't typically always follow a 24-hour circadian rhythm like humans do. They have been shown to follow longer or even shorter. I think there is evidence that there's like even some of the shortest circadian rhythms have been shown. So, and because of this, we are looking to study how wolf spiders across different temporal niches, whether they're nocturnal or diurnal, meaning like active during the day or active during the night, compare in their metabolic rate or respiration relative to those natural circadian rhythms. All right, so in terms of our methods, Katie, I might just kind of echo what Katie kind of talked about, but we collected at night and we also collected during the day. This is a picture of a night collection at a local field site, and we collected across different species, mainly being schizocosa, pardosa, rabidosa, and after collection, they were all fed flies, and we brought them into our lab and consequently put them in a 14 and 8-hour light-dark cycle to mimic environmental conditions with a ramp period to account for dawn and dusk. And prior to our functional response data recording, that was when we took our respirometry data in 3 different temporal conditions, which were afternoon, twilight, and evening. So we took On the right, you can see the equipment that we use to measure the respiration of all the individual wolf spiders. These pre-sense sensors have, each individual vial has a sensor at the bottom that's it is able to record changes in oxygen concentration and pH concentration. And so that was a way we could record the metabolic rates of the individual wolf spiders. And this was all done as we were taking each of the concentration values and data. We tried to mimic as close of a dark environment as possible by wrapping aluminum foil around each of the vials and turning off like all external light, just to eliminate any effects of like external light. Let's see. Okay. So once the data was taken, we had to obviously analyze the data. So doing so, I used Excel, and basically we had just a bunch of values across time for each of the individual spiders in each vial, and we had to eliminate the first like 20 to 25 minutes just for a calibration and system equilibration, and then we took the values at every 2 minutes for about like an hour. So using that green line, just the green highlighted, just kind of shows where we started. Using each spider's mass, we were able to take their individual spider volume in order to account for the fact that in each, obviously we had variation across the sizes of each spider. So some of the spiders were really cramped in there, some of them had a lot of space. And that means some had, the oxygen concentration levels are then gonna have to account for the fact that like they vary in size in these vials. So doing so, we use the spider volume and as well as the volume of the vial in order to calibrate, and we used 2 control vials in each set of each set of data that we took at the different temporal conditions. And we looked at the metabolic rate just across the different spider species, and then I was able to graph just across time the different oxygen values or respiration values. And then at the top you can kind of just see for each spider they had 3 different respiration values, one at afternoon, evening, and twilight. So this is just sort of an overview of the sample size or population that we took respiration values of, and these are, as you can see below, these are the different species and across sex and when they were collected. So in total, it was about like 60 spiders that we ended up taking respirometry data from. So these were the graphs that we were able to, box plot graphs that we were able to produce using those respirometry data, and this is across sex. So you see the F and the M representing the different females and males across the different temporal conditions, and the variation that we see for the afternoon female and afternoon male. This is across temporal niche, as in when they were collected.
Can you explain the respiration axis to me briefly?
Yeah, so this is like the respiration that we have, or like the values. Yeah, so this is like the slope values of when we graphed across time the oxygen concentration values. So that's why you see like close to zero for a lot of them, because a lot of them it's close to zero. I don't know if I'm explaining it best.
You're right there. So the basic idea, right, is you put anything in a tube, and you close that tube, it's going to start burning that oxygen, right, and converting that oxygen into carbon dioxide. And so you can measure the amount of oxygen concentration over time, and then you get, you should get this negative line, and then you can plot the slope. And so what she's showing here is the slope. Now, anyone that understood what I just said will say, well, why is there one with a value of 10?
Huge
and that's
variation.
because some of these data got very weird because we did an environmental chamber where the pressure changed. So we're still working through that. So the value of like 11 there, and then anything less than negative 10 is weird too, so just ignore that. But we were working on these data last night, so, so we haven't really cleaned them all up yet.
So like obviously
that's what you're seeing.
Close to like negative 15 is kind of raises some eyebrows, but as Brett was talking about, there's definitely some considerations. Obviously we tried our best to account for the size differences, but we also saw some weird like cycling that was happening. So in order, due to the pressure changes in the chamber, so that's something we will continue to have to work on for sure as we work through this data. But due to time constraints, unfortunately, that's not been possible. And this is just across the different times when we took them, like afternoon, evening, and twilight. So again, considering all the constraints that we had with like the cycling, there were some findings in that like the afternoon had significantly more variation, like just the bigger box plot, and during the day, wolf spiders that were collected at night had the most variation compared to other temporal niches, males and females, and the afternoon light conditions had the most variation, with twilight having the least. And some significance of this is just that, as we know, metabolism and circadian rhythm can vary for the same organism throughout the same day, just even depending on what time of the day it is. And it's really important because light pollution is becoming an important, significant issue, and if we do have, continued artificial light, what does that mean for individual organisms, for arthropods, and for their metabolism? And just some thank yous for funding and Stella for providing equipment and resources, and yeah.
Are there any questions?
I've got one.
Yes.
So can you go back to the slides? Let's see. Wait, I wanted the one where it was nope. Go back, right here.
Okay, cool.
Okay, cool. So can you run us through really quick what this is showing? So what are these data? Like, not worrying about the respiration, just who's in the afternoon, who's in the evening, who's in the twilight. What's this showing?
Well, so each, this is showing like all the individual organisms, because each organism had data taken at afternoon time, evening time, and twilight. And so this is just showing, I guess, the variation of between the afternoon values that we have for all the organisms, and then evening, and then twilight.
Yeah, so this is really cool, right? Because again, these are all the same individuals. So all the individuals were measured at afternoon, all the individuals were measured at evening, and all the individuals were measured at twilight. So knowing that, right? It's not like we had 3 different groups. These are all the same individual. So what do you take from this? And again, it's noisy. But when you look at this figure, this is showing something that I think is pretty cool. And yeah, so what do you see about respiration across these 60
It's
spiders?
a lot more active across, during the afternoon we see like just how, you know, more
Yeah, and
active it is
respiration.
as compared to like
Right.
the twilight.
And the respiration's bigger, right? So
Yeah.
a greater negative value of respiration means
That means like
animals.
some are like have a lot higher respiration and some
Yeah.
are not at all, which can linking back to the fact that we know there's you know different temporal niches of like night and like are when they were collected at night and when they were collected at day, possibly meaning like nocturnal and diurnal. This can maybe account for the fact that like some are more active during the day and some are less active during the
Yes,
day.
this is interesting though in the sense of what it, you know, with Katie's data
Yeah,
showing that most were active at twilight.
yeah.
Is this what you'd expect to see for respiration? Or is this the opposite?
No, this is the opposite for
Right.
sure.
Okay,
Yeah.
Ben, take us away, man.
So, I think I'm following all. Does this look different from, like,
What
have you
you
guys
would
found that this
expect.
looks different from one day to the next to the next? Because from my understanding, like, if you had like a 16-hour circadian
Mhm.
rhythm, then they might be more active, like, early in the morning if they've been early, or really active in the afternoon. So is that kind of,
Yeah, so well, we took data from I think 2 different days, and we haven't yet compared between those 2 different days, but that'd be definitely interesting to know any differences across those 2 days of like how this compares. But yeah. Yeah.
Could this be viewed as like a suggestion as to how they're going to be knowing when it's time to go into their winter dormancy as like the days shorten and stuff? And their respiration is going to kind of step up more so.
Possibly. I think with winter dormancy, obviously temperature is like a huge factor, and we just didn't look at that in relative to like the metabolism and the rates of respiration. But I think that'd be really interesting to look at about how that might cue into, you know, it's time to hibernate or time to, you know, use less energy during the winter. But yeah, good question. Okay, cool.